just setting up the YouTube stream. Okay. Yeah, so as many of you will know, in case it's your first time here, this is a part of the Brain-Body Interaction seminar series, which is more or less weekly series that we've been running on the theme of brain-body interactions, covering all sorts of topics from how the brain interacts with the immune system to breathing and physiology, and also in terms of psychiatry, as we've seen in the last couple of seminars that we have had. So before I introduce this week's speaker, just a couple of housekeeping things. So if you want to chat during the seminar, you have the chat window on the right. And if you want to ask a question, use the Ask a Question tab at the bottom of the screen. And if you want to read your question out loud at the end of the seminar, just put on screen or something at the end of your question, and then I'll invite you on screen to ask the question. Otherwise, I'll read it out at the end. And yeah, we'll also have a smaller kind of meet the speaker session after the seminar. You will have been prompted to join that, to sign up for that when you join the crowdcast. But in case you missed it, feel free to send us an email. I'll put our email address in the chat window, and you can send an email to sign up for that. So without further ado, I'll introduce this week's speaker. So we're really excited this week to welcome Kara Marshall, who is a postdoc in Adam Pataputian's lab at Scripps in California. So Kara did her PhD initially in Texas, and then moved to Columbia with her supervisor, Ellen Lumpkin. And there she studied touch receptors in mammals in all sorts of different contexts. So, for example, she looked at the features of touch receptors in the skin, their structure, and how they're remodeled when they undergo rapid remodeling. She also looked at somatosensory neurons that innervate the wings of bats, and looked at trip expression in sensory neurons that innervate the pancreas, actually, which is a really interesting aspect of brain-body interactions. And she really continued this theme of looking at mechanosensory receptors in different contexts when she moved to a postdoc in the Pataputian lab. And so she's done some really fantastic work looking at the role of piezo ion channels in sensing filling of the bladder, and how that contributes to the control of urination in mice and in humans. And she's also contributed strongly to work looking at the role of piezo ion channels in sensing blood pressure, and also in the innervation of the gastrointestinal tract and how that contributes to feeding and digestion. So I hope we're going to hear about some of this exciting work today. We're really happy to have you here, and looking forward to talking about it.
Great. Thank you so much for the introduction. I'll share my screen. Yeah, I'm really excited to be here and excited to know about this series. So hopefully you can all see my slides. So yeah, as was mentioned, I'm really interested in mechanotransduction, and this started when I studied touch receptors in the skin, but now I've moved inside the body. And so I'm going to tell you a little bit about a couple different stories, some of which are published, a little bit of unpublished work on that today. So when we think of our senses, we tend to think of these 5 senses as defined by Aristotle. And these are our exteroceptive senses, so how we take in information from the outside world. And so, that might be through photons or chemicals. But I'm most interested in this mechanical aspect, which I'd say is really the least understood of all of these senses, is how we sense mechanical force. But this is also just a very limited view of what our senses can do. This is what we're kind of most familiar with, but there's actually a number of senses that are interoceptive senses. So one of these is when we generate force from our own bodies by stretching our muscles, in the case of proprioception. But then there's a whole host of senses that are occurring, beneath our level of consciousness in our internal organs. And these are both chemical and mechanical senses. And these are really critical to various organ functions, like our sense of blood pressure, digestion, peristalsis through our gut is mediated by mechanosensing, as well as bladder filling. And so these kind of are running without our conscious, perception most of the time. But of course, if something goes wrong with one of these senses, it can really throw off the function of a given organ. So it's important that we understand a lot more about how these interoceptive senses work. And I guess this audience is actually all right in line with that and cares about these things too. So, excuse me. So I also wanted to mention that mechanotransduction, while it's the least understood of these senses, is actually really universal. For instance, the first mechanotransduction ion channel was discovered in bacteria. These are MISC channels. And bacteria use it as sort of an osmotic pressure release valve. So if they're in a hypotonic solution, they need to release solutes to not burst. They use this MISC channel to do that. So all the way up from bacteria, we found similar, ion channels, not we, but the broader community found similar ion channels in plants, because plants sense gravity and also can respond to damage to their leaves. And then there are other ion channel families found in worms and flies. And for a long time in the field, people were studying all of these different ion channel families and actually trying to connect them back to a homologue in mammals. But for a very long time, it was not known what the primary mechanosensors were in mammals. And the reason I'm taking you through all of this is because really, without understanding the ion channel or the mechanotransduction channel, it's very difficult to understand its role in physiology. And so this remained a big mystery in the field for a long time. And so our big question was really, how do mammals detect force? And this is something my lab, the Patapudian lab, made progress on about 10 years ago. So Bertrand Coste in lab discovered a family of ion channels called Piezo ion channels. He did this by actually just poking cells in a dish. And he found cells that were inherently mechanosensitive in that when he poked them, they had inward current. And then, through a very long process of knocking down genes one by one, he narrowed it down to what he termed piezo ion channels, as FAM38A. And really, this meets the key criterion for being a true mechanosensor, in that he can overexpress it in a mechanically silent cell and actually confer mechanosensitivity, such that with increasing levels of force, he gets increasing currents. So this was very exciting for the field. And so there are actually two of these ion channels, Piezo1 and Piezo2. And there are homologues in all over different kingdoms, so in invertebrates, plants, and even in slime molds. And then most exciting for me, it was Piezo ion channels were expressed in many different mammalian tissues. And my favorite is the nervous system. So just as a reminder, for those who aren't as familiar, the sensory nervous system cell bodies actually reside in little clusters of neurons called dorsal root ganglia, which sit next to the spine. So if you look at an organ like the skin, for instance, you can see in cross-section many different ending types with different morphologies to detect varied stimuli. But these are just the ends of the cells, and the cell bodies are all the way back here. And if I were to look at a cross-section, it looks like this, where you have this cell body that sends projections centrally and out to the periphery. So if I look at, say, a cross-section of a mouse, for instance, it would look something like this, where all of these ganglia actually send innervation to the skin and the muscle and to internal organs. And so when you look at a cross-section of DRG in situ, and you can see that there's a ton of Piezo2 expression in these neurons. And so when this was first discovered long ago, of course, the first question is, given that they're all over, Piezo2 is all over the sensory neurons, what are they doing in terms of mechanosensation? And so I'm briefly summarizing a lot of work from lab that found Piezo2 is really the primary sensor for touch and proprioception. So just as an example, this is what muscle fires. This is a muscle firing in response to stretch. And you can see that without Piezo2, the muscle is mostly silent. So this means that, without understanding muscle stretch or without sensing muscle stretch, really you have no idea where your limbs are in space. And so when we look at an animal that actually lacks Piezo2 in its caudal tissues, this is what we see. And you can tell that, this is not a normal mouse locomotion. So this mouse, because it has no proprioception in its hind limbs, it also really has no idea where its limbs are. And so it's in all of these strange positions, and its tail goes all over the place. So it's a pretty profound phenotype. And it just goes to show how important proprioception is for even just understanding, where our body should be at any given time. And so, without these critical senses, it really changes our interactions with the world. And so at this point, when I came into lab, the role for Piezo2 and touch and proprioception had been defined. And I really wanted to make headway in understanding what these ion channels could be doing for interoceptive senses. So I'm going to tell you briefly about 3 stories today. The first is on the bladder, which is published recently. And then I'll tell you a little bit about an older story I contributed to on baroreception, which is blood pressure sensing, and to lead into a little bit of new data on the stomach. So first, we'll talk about bladder fullness. And as I mentioned, this is a story that came out recently on the role of Piezo2 in urination. So why study urination? I mean, this is something, obviously, we all do it every day, and it's really important to you, whether you realize it or not, because a lot of people end up having issues with urination. And this is something that, there are so many different things that can go wrong. And when something does go wrong, it greatly impacts your quality of life, whether that be, overactive bladder or underactive bladder or incontinence. So it's really critically important that we know exactly how the system works if we want to better develop therapies for these many issues that can arise. And then for me, it's a really interesting system because unlike a lot of interoceptive systems, it relies mostly on mechanical cues for its normal function, whether that cue be bladder stretch or fluid flow. And then lastly, there's a lot of big questions in this field still, because we did not know the molecules that mediate mechanotransduction, nor did we know which cell types were most important. And so because of this, we really didn't understand what the consequences were when mechanosensation was impaired. And so I set out to understand this problem. So just to cover how you urinate, you have a muscle around your bladder called the detrusor muscle. This is smooth muscle. And then you also have sphincter muscles in your urethra. And these 2 muscle groups have to coordinate for efficient urination. So when you sense bladder stretch, that actually initiates mechanical reflexes that induce relaxation of the urethra. And vice versa, when you sense fluid flow or stretch in your urethra, that sends reflexes back to the bladder to increase contraction. So just imagine if you're trying to contract your bladder, but your urethra is not relaxed, nothing will come out. Similarly, if you relax your urethra but don't contract the bladder, it's really inefficient. So this is why I'll be telling you about the activity of both of these muscle groups together, because this coordination is critically important. And then, as I mentioned previously, all of the signals that initiate these reflexes are mechanical in nature. So I wanted to ask 3 big questions. Is Piezo2 involved in urination? And then what are the functions of mechanosensation in the system? And what cell types govern mechanosensation? So first, I wanted to know whether Piezo2 was necessary for proper urinary reflexes. And to do this, I set up cystometry, where I put saline-filled lines into the bladder, connected to a syringe pump so I can control fluid filling, and a pressure sensor so I can read out pressure of the system. And over 20 minutes, this is the kind of recording you get where as I slowly fill the bladder, it reaches a threshold, and that means it's time to urinate, and it undergoes this Micturition cycle. And so with each peak, this is the bladder contracting back on the saline lines. And so this would be equivalent to a urination event. At the same time, I have EMG electrodes in the urethra, and you can see this really nice coordinated burst of activity from the urethra. And again, this is actually helping to get the urine through. In male mice, it's actually, the urethra acts sort of as a pump because it's such a long, narrow tube. And so these 2 muscle groups are intimately coordinated. So if we look at a wild type animal, these are 3 different animals actually, you can see that they have very regular contraction intervals because I'm putting the same continual fluid flow through each of these. But if I look at an animal that lacks all Piezo2 in their caudal tissues, this is the animal I showed you before that has the proprioceptive deficits, you actually see something very different. So not only do they have irregular intervals, and sometimes they have these long plateaus leading up to their urination event, but sometimes they have very long intervals between contractions, indicating it takes more fluid for them to actually initiate a urinary event, or they're less sensitive. So if I look at just the time between contractions, you can see that it's longer for knockout animals. And so overall, this indicates that mice without Piezo2 actually have higher bladder stretch thresholds. So it takes more to initiate one of these urination events. But I also wanted to know, once the event is initiated, is it sustained normally? And so again, to do this, I'm actually just looking at individual events separated out, and I'm putting all of these events together in a single plot. And I'll show, if I zoom in, I'll show an individual pressure contraction event that you see here with a heat map, where red indicates high pressure and blue is low pressure. You see that this pressure event has this sort of double humped appearance, and that's because, as the bladder starts to contract, when the urethra activity kicks in, it starts urination, and that relieves some of the pressure, and that's kind of why you get this double hump, because the bladder is still contracting, pressure gets relieved, and it continues to contract. So at the same time, I'll also show urethra EMG, and that looks like this. And you can see this bursting that I mentioned in the male urethra, where it actually, it kind of fires repetitively to help get urine through. And I'll also show that as, black and white events with black indicating high activity. So if we look at just one of these heat maps, which indicates, this has 6 mice with all of their contraction events piled together. So an individual line is an individual contraction event. These arrows indicate the break between one mouse and the next. You can see that in wild-type animals, these events are very regular. And between mice, it's slightly different. This mouse has smaller contraction events, for instance. But overall, these are very regular events. But it looks quite different in knockout animals. And so you can see here, you have a lot of irregularity between their contraction events, and some of them have very high pressures leading up to the contraction. So if I look at just the area under the curve, which means really how much pressure is required to urinate, you see that knockout animals have require much more pressure. At the same time, if I look at urethra EMG, you can see that there's this really nicely coordinated burst of activity underneath the bladder peaks, the bladder pressure peaks. But this is mostly gone in knockout animals. Or if it's there, it's actually just not coordinated as well. And if I look at just total activity, you can see that it's greatly attenuated in knockout animals. So this indicates that, I wanted to also point out there's a lot of zeros there that might be hard to see. So really this indicates that Piezo2 is required for appropriate reflexes and coordination that those reflexes enable. So there are consequences if these reflexes aren't working properly. And this is an example in humans whereby, at first, if you can't empty your bladder, there's compensation. So the muscle grows, there's hypertrophy to actually overcome this difficulty. But over time, that leads to what's called decompensation, where this muscle remodeling becomes really damaging to the organ. And it can actually eventually end in organ failure. So it's really dangerous if these problems continue. And so I wanted to know if in these mice, which lack appropriate reflexes, if this actually occurs. And so when I look at just a cross-section of the bladder, you can see on the left, wild-type mice, the urothelium is this top layer, the middle layer is the detrusor muscle. And in knockout mice, they do show really remarkable remodeling. And these are age-matched controls, but I can actually measure this just by weighing the bladder. If I just take it out, weigh the freshly excised weight, you can see that the knockout animals are much heavier, indicating they've undergone remodeling. And so lastly, I wanted to understand if this changes the way that they urinate, their behavior. And so mice have a very stereotyped pattern of urination. You might think that only humans care about where they urinate, but that's not true. Anyone who has a dog knows that they care really deeply about where and when they go to the bathroom. And mice do too. So if you put female mice just in a regular cage on filter paper, they tend to urinate in the corners and at the edges. and not so much in the middle, which is more dangerous. Of course, male mice will territorially mark, and so I'm actually just using female mice here. But when I put a knockout animal on the paper, you can see that the pattern is really different. So sometimes they actually urinate in the middle of the cage. And you can see this with several examples here. These are 5 different wild-type mice. Again, they're very consistent. They go on the edges and the corners. But with knockout mice, it's highly varied. So sometimes you get these small urinary events like leaks, so indicating they have some incontinence. And then sometimes, as I mentioned before, they'll just urinate in the middle of the cage, which really indicates a lack of volitional control, as if they aren't able to go to a corner or an edge to plan out their urination. And so if I look at just the percent of urination in the middle of the cage, it's much higher. It's not necessarily because they spend more time in the middle of the cage. So this indicates that maybe these mice lack some element of volitional control. And so, everything I've shown you until now has really been the motor output of this system. But ultimately, I care about sensation. And so we really wanted to know whether Piezo2 itself was the sensor for bladder stretch. And to do this, we had to look in sensory neurons themselves. And so I teamed up with Nima Gitani in the Chesler lab to do DRG imaging, where we inject cream mice and pups. And when they're adults, this means that it excises stop codon for GCAMP, and it's a Piezo2 floxed mouse. So any cell that expresses GCAMP will also be a Piezo2 knockout cell, and then controls are just GCAMP alone. And so after this pre-injection, we waited till they were adults, and then I performed my cystometry setup by catheterizing the bladder. And then we turned it over, and Nima exposed this first sacral DRG. Now, this is something that I always like to say, this is not done very often. And like people who study the CNS, they do imaging of neurons all the time in response to specific stimuli, but it's much more difficult, and we're kind of a little bit behind in the peripheral nervous system. So we were really excited when we were able to actually see in response to pressure, which I show, you can see at the bottom. When I fill the bladder, you can see this really nice activation of a number of neurons. So this was the first time anyone had seen this, and it was very exciting for us. So if we look at wild-type animals, we'll see a number of cells light up in response to bladder stretch. But in knockout animals, we saw many fewer cells. In fact, this is the best example I found with the most responders. Most of the time, it was a black rectangle. So this also revealed some new biology for us, which was exciting, because we found that in wild-type animals, we had cells responding to low and high pressure, but only the low pressure cells were missing in the knockout animals, indicating Piezo2 is responsible for low threshold bladder stretch sensing, but also that there's another sensor that does high threshold. And so just to give you a sense of this through traces, on the top are pressure traces, and this is me manually filling the bladder. You can see the average calcium response below, but again, you can see at these lower pressures, all the calcium responses are missing in knockout animals. So our next question is, which cell types are really critical for this mechanosensation? And one of the exciting things that indicated it might not just be neurons is that when I looked in the urothelium, the inner lining of the bladder, at these specialized cells called umbrella cells, which are marked by keratin 20 in green here, I did actually see Piezo2 transcript. And this is interesting because one of the prevailing ideas in the field was actually that stretch put pressure or stretched out the urothelium itself, and that the urothelial cells released something to activate neurons below. In fact, most people think this is ATP. And so we really wanted to know whether it was the urothelial cells that were sensing stretch or the neurons. And so just as a reminder, if these are 3 examples of what wild-type cystometry looks like. Again, really regular contractions with very regular coordinated urethral responses. And I'm just going to show you the qualitative data from these animals to give you a sense of what's different. So I was able to separate out the function of these cell types by looking at just a neuronal knockout and comparing to a urothelial Piezo2 knockout. And so you see that in the neuronal knockout, we have these long gaps between contractions, as we saw before, as well as really uncoordinated urethral response. But I was really amazed when I looked at the urothelial knockout and saw something very similar, indicating that really both of these cell types are working together and using Piezo2 to coordinate these mechanosensory reflexes. And so ultimately, we think we have a lot of evidence that Piezo2 is involved in urination, very critical. for low threshold bladder stretch sensing, Micturition reflex sensitivity, as well as voluntary voiding. And interestingly, it puts forth this two-cell model of mechanosensation. Both urothelial cells and their sensory neurons are working together to sense stretch. And this is actually an emerging theme in the body because there's a lot of mechanosensors that do this. So I actually used to study Merkel cell neurite complexes in the skin. And this is where you have specialized epithelial cells called Merkel cells that express Piezo2 and actually coordinate and shape the response of Piezo2 positive neurons beneath them. And there's similar arrangements in the lung with neuroepithelial bodies, as well as in the gut with enterochromaffin cells. So I think that this system is really fascinating, and actually it can reveal a lot more about other systems in the body that work together with specialized epithelial cells and neurons. But lastly, we wanted to ask the question that, I think all of us who care about mammalian physiology always would love to ask, which, is this relevant in humans? And so we were really fortunate to team up with Alex Chesler and Karsten Bonneman at the NIH, who had done a really lovely job characterizing a group of patients that were Piezo2 deficient. And so they found, this was back in 2016, they found that these people had very limited gentle touch sensation, and they also had no proprioception. And so this paper was really landmark for the field because, we had found this in mice, but to confirm that humans, the function of Piezo2 is conserved in humans was a really, really amazing finding. And it really helps us understand a lot more about where Piezo2 is functioning in the system. So just as an example of what it looks like when someone lacks proprioception, this patient was asked simply to walk in a straight line, but she was given, an eye cover, so she had no visual cues, and a harness to protect her. And you can see that this is very difficult for her. And this is because she's blind to her own body without proprioception. She doesn't know where her limbs are without visual cues. And the really amazing thing is that so many of these patients have learned to compensate for these deficits by using visual cues to move through the world, which, as you can imagine, seems very difficult. And so these patients were just incredibly generous with their time. And they're, in working with Alex and Karsten and Dima Saad at the NIH. And they're able to offer so much more than what an animal model can offer, and that, they offer their stories. And so one of the stories they told is about urination. And we set out to characterize the deficits that they have in urinary control, because they actually mentioned in their interviews that they didn't really ever feel the need to go to the bathroom. And so when we set out to characterize this better, the most remarkable finding is that they only urinated 1 or 2 times per day, which is highly abnormal. A normal response, or a normal frequency, would be 5 or 6 times per day. And this is because they just scheduled their urination. Since they never felt the need to go, they scheduled it so that they didn't have an accident. This is actually also a technique employed by parents. Many of these patients were children, so the parents actually adopted a schedule for them. And so I'll show you a little bit. We sent them a questionnaire, and I'll show you a little bit about some of the answers on this questionnaire to better characterize their deficits in urinary function. You can see, I'm sorry, that's cut off a little bit. You can see the patients vary in age. And I'm going to show you their answers to these questions with gray indicating a normal response and any colored box indicating some abnormal or pathological response. And so many said that they have to rush to the washroom to pee, or when they pee, it stops and starts. And this is really indicative of abnormal reflexes, as I mentioned before, because they're not able to plan when they're going to the bathroom, and they're not able to sustain urination normally. And also, almost every patient said that they have to push or wait for their pee to start. And in fact, the one who didn't say this was a 9-year-old, and she hadn't achieved continence yet. So interestingly, some of the adults independently told us that they use their hands to push on their abdomen in order to empty completely. And so this really indicates a profound deficit in their urinary control. And this was a really good indication that the function of Piezo2 was conserved in humans. So we're really grateful to those patients for actually coming in and giving us all that information. There are still, of course, really big open questions in this field. Namely, what are the other mechanosensors functioning in the lower urinary tract? As I mentioned, we know that high threshold sensing is not mediated by Piezo2. And in fact, the humans, they didn't feel the need to go to the bathroom, and they couldn't feel this fullness that you or I could feel, where you can plan ahead, right, and say, oh, I might need to get to the bathroom in 20 minutes or so. But when it was urgent, they could feel it. So it was almost like by the time they felt it, it was too late, again, indicating there's a high pressure sensor that's left. Of course, there's really huge questions about how these urothelial cells coordinate and communicate with the neurons below them. And I'm also interested in the role of these mechanosensors in painful pathologies like cystitis, which actually affects a large number of women every year. And it would be interesting to understand whether or not these sensors contribute. So this was one interesting story about how Piezo2 is required for interoception, but everything I told you up till now has been DRGs. And there's actually a lot of innervation of your internal organs mediated by other tissues or cranial ganglia. And so I'll tell you about a couple of those right now. So I'll start with this story on baroreception or blood pressure sensing. And as I mentioned, blood pressure sensing, gastrointestinal innervation, these are all mediated by the Nodose ganglia innervation. So DRGs have Piezo2, but Nodose ganglia, which sit at the base of the skull, actually have Piezo1 and 2 positive cells. And so the Nodose ganglia supplies innervation to the vagus nerve, which really travels throughout the body and innervates most major organs. And so this story was really primarily the work of Wei Zhang, who is now starting his own lab. And this is, this goes back to something that we're all familiar with in the doctor's office, because every time we've gone to the doctor, they've probably taken, our blood pressure. And this is because it's one of sort of the basic vital signs. And this is because it's incredibly important to your health. And so this is mediated in part by the baroreflex. So you have both long-term and short-term mechanisms to control your blood pressure. The baroreflex is this very fast-acting homeostatic negative feedback loop. And so the role of this is to maintain a constant mean arterial blood pressure. And so that means that when you stand up, you generally will lose pressure dramatically, right? And the only reason you don't faint is because of the baroreflex. Because as soon as the stretch receptors in your aorta and in your carotid sinus detect this, rapid decrease in blood pressure, they send signals to your vasculature to actually contract to keep blood in the head, and they speed up your heart rate. Similarly, if you were to, have really high blood pressure, the stretch reflex would actually indicate that, vasodilation should occur and your heart rate should slow down. So it feeds back on itself to maintain a constant pressure. And if this is compromised, this leads to arrhythmias and hypertension and heart failure. So this is a really important feedback loop. And so the neurons that mediate this are really beautiful, and they were discovered a long time ago. This reflex has been characterized for 100 years. And you can see this beautiful drawing of these endings on the left. And then on the right, in a picture I took, you can actually see the architecture of this neuron, or these neurons, which actually wrap a ring around the central portion of the aorta where it's sensing stretch. So with each heartbeat, the aorta is stretching, and that's the stimulus for these neurons. And so despite knowing about the reflex and knowing about the neuron endings for years, it wasn't known what the sensor was. And this is what we set out to identify. There were other ion channels that had been identified as contributing to the baroreflex, but none that were really wholly responsible for it. And so to study this, we looked at Piezo2 and Piezo1 double knockout mice using FOX2B Cree animal. So these are essentially mechanically silent in their Nodose ganglion. And data I'm not showing Wei Zhang has actually recorded and found very few mechanical responses left in this ganglion. And so the way Wei Zhang studies this is by using a drug, phenylephrine, to induce the baroreflex. So what this looks like is the blood pressure when he injects phenylephrine, which acts only on the vasculature, not on anything in the heart, actually shoots up. And again, because of the baroreflex, what you see is this compensatory decrease in heart rate to compensate. But when he looks at animals that lack Piezo1 and Piezo2 in their Nodose ganglion, this decrease in heart rate is gone, indicating the baroreflex is absent. And I remember when he first started studying this, at first he was really disappointed, because when he looked and measured the baroreflex in individual knockout mice, Piezo1 or Piezo2 alone knockout mice, the mice were fine. The barrier reflex was normal. It was only the double knockouts where he sees a phenotype. So this indicates that Piezo1 and 2 actually work together. And this was the first time we'd seen this, because as I mentioned in all the stories previously, it was mostly actually Piezo2 that was really functioning in the nervous system alone. But here they work together. And so he also found that these animals, these knockout animals, have higher blood pressure, which actually corresponds with the humans who lack a barrier reflex. So ultimately, piezos mediate neuronal sensing of blood pressure. And in data I'm not showing you, if you activate Piezo2 positive neurons, this is sufficient to induce the barrier reflex optogenetically. And so this was a really exciting story because it was, unveiled like the first time Piezo1 and 2 are working together. And it's also, uncovered a really long-standing mystery about what's mediating this stretch reflex. But that also indicates there are probably other interoceptive processes in the Nodose, or that are mediated by the Nodose ganglion neurons that depend on piezo ion channels. And so to round this out, I'm going to briefly tell you about a little bit of work I'm doing on understanding the role of gastrointestinal stretch in satiety. Because we all know anecdotally that, that when you eat a large meal, you feel really, really full. And this actually is supposed to actually stop you from eating more, although it doesn't always work. And so I'm really interested in understanding in the stomach and the gastrointestinal tract how this stretch reflex could contribute to feeding behaviors. And so the way you sense satiety is complex. There's obviously a lot of different contributing factors. Of course, you have hormones and innervation in your hormones coming from your periphery and innervation in your periphery that sends signals back to the brainstem and the central nervous system and into places like the arcuate and the parabrachial nucleus that control feeding. And I'm obviously focusing on vagal afferents today, not hormones, because these are much less understood, I would say. And coming from the gastrointestinal tract, you have both mechanical and nutrient cues. And I'll focus just on mechanical cues. What I hope to understand is, what are the mechanosensory proteins that detect gastrointestinal stretch? And also, how does this mechanosensing influence physiology? And this goes back to what I mentioned at the beginning, which if we don't have the sensors, if we don't understand the molecules that are functioning in the system, it's very difficult to understand what the role of mechanosensation is in physiology. And so that's the whole goal. And so first I wanted to understand whether mechanoreceptor activity changed with just normal feeding. And to do this, I used a dye that labels mechanoreceptors. And I put animals on an overnight fast, injected this dye intraperitoneally, and then either continued the fast with one group or refed them. And then I imaged the tissue in whole mount. And so you can see that this works well to cause stretch in the stomach. This fasted stomach is much smaller than the fed stomach. The fasted stomach is not completely empty because it turns out mice do actually still eat their poop, which is kind of gross. But of course, this works really well to stretch the stomach with normal feeding. And if you look at the stomach weight, you can see the contents are much higher in the fed animals. And so if I look at this label of mechanoreceptors, FM143, in whole mount, what I see is that there's much brighter labeling in the intraganglionic laminar endings of the stomach. So these are endings that intercalate with the myenteric plexus in the stomach. And these are thought to be stretch sensitive. And you can see that just with labeling alone, it indicates that maybe these neurons are actually more active during feeding. And so this was a good indication that, maybe piezo was functioning in the system just to, and during normal feeding. But ultimately we want to understand if they eat differently, right? So in animals that lack both Piezo1 and 2, we put them into a clams machine, which really measures all of their food intake over multiple days. And we see a difference in what they eat during the light phase. Interestingly, you can see there's huge variation in what mice eat. Males are in blue, females are in purple. But ultimately, if we look only during the light phase, we see that knockout animals do eat more, indicating maybe they lack this stretch reflex or stretch sense that inhibits eating. It wasn't because they ate bigger meals. It was actually that they ate more meals. And this goes back to this classic idea of satiety, which is not so much how much you're eating right then, it's how long before you eat your next meal. And so we think maybe piezos are playing a role in satiety, but there's a lot more work to do in this area. So, I'd like to know, better understand what stimuli these mechanosensors respond to. And lastly, because there's expression of Piezo1 and Piezo2 in the Nodose ganglion, we don't know what the distinct roles for these proteins are. And they're, as I showed you, they're actually in different cell types. And so maybe these different cell types are mediating different kinds of, or sensing different kinds of stimuli. And lastly, I really am interested in understanding what happens when you have too much mechanosensation in this system. And that's because there's actually a number of alleles, both gain of function and loss of function, that we found in humans. And one example of this is this E756 deletion, which we found in human populations. And I'm not going to tell you about this story today, but it is a really fascinating story from my colleague Shang Ma, who's currently on the job market, whereby he found that about 1/3 of all people of African descent have a gain of function allele in Piezo1. And remarkably, this seems to be protective against malaria because Piezo1 is expressed in red blood cells. And this is one of these really exciting findings where, we didn't expect really that Piezo1 would be in red blood cells or that red blood cells would even need a mechanosensor. But it seems that evolutionarily, this has been selected for because it's protective against malaria. And so given that 1/3 of all people of African descent have this allele, this gain of function allele, and Piezo1 is actually expressed all over the body, it's expressed in many different tissues, I'm really curious about what other elements of physiology it might affect. And so this is sort of an interesting future direction for the lab. Oh, and I showed of the actual numbers are below. So you can see 400 million people have this phenotype, or this genotype. So, there's a lot of questions in the system that I'm eager to ask. And, in defining sort of the future of, my lab, which will be starting in a year or so, I'm really thinking about these questions in two parts, both what is the peripheral nervous system doing and then what is the central nervous system doing? Because we still have a lot of, open questions in the peripheral nervous system, namely, what are the mechanosensors and what are the, cellular identities of mechanosensors that function in both healthy states and in diseased states. So, this is just the first step. By understanding that piezos are important in these systems, we can also understand how they contribute to pain states, for instance. And I really think the bladder and its innervating neurons are a great system to do this because you have both Piezo1 and Piezo2 positive cells in the bladder. I didn't show you all of those data. And we know that they're communicating in some way with innervating sensory neurons. So I think just by understanding these peripheral mechanisms, we can learn a lot about this system and others. And then I also really want to understand, ultimately what's happening in the brain. Because to understand interoception, we have to understand how it's encoded centrally. And so I'd love to know more about what circuits are engaged by internal mechanical stimuli, and then how these control physiology. And I really think the gastrointestinal tract will be a great system to study this, because we know so much about the central circuits that control feeding. For instance, the cell types in the arcuate, when they're active, and what they're mediating. And so I think by linking the peripheral nervous system into known circuits in the CNS, we'll actually be able to understand a lot more about how this system functions. So that's the future, and I'm happy to talk more about that. I also wanted to thank my wonderful lab members, in particular our DEM, for being a great mentor, and then a lot of wonderful collaborators in lab. And Wei Zhang, as I mentioned, really pioneered the blood pressure study, and then many others have helped with the other studies I showed you. And then we have just amazing collaborators at the NIH in the Chesler lab and the Boneman lab. In particular, Nima helped a lot to get that DRG imaging working. And then Jason Keller, who was a student in the Stowers lab next door, actually they got us up and running on all the urinary tract function stuff. So I'm really forever grateful to them. And it turns out I've spoken very fast. I'm saying it's only 8:45, but that's good because maybe we'll have time for questions. So anything I ran through too fast, feel free to ask me about.
Yeah, that's great. We have plenty of time for questions. There's a really fantastic talk. There's some really fascinating ideas there and some definitely things that will stimulate discussion here. So I'll start off with a question, actually. So I was really interested in your findings about in the bladder with the necessity of Piezo2 in both the urothelial cells and the neurons. And you mentioned that that might be a theme that's also the case in the GI tract. So I was wondering if you could talk a bit more about that, like what are the epithelial cells that express piezo ion channels, and do you think they might interact with IGOs or some other vagal efferents?
Well, so I can't speak as much to the gastrointestinal tract. Certainly there are enterochromaffin cells that express Piezo2. Art Vader has done some work, and others. And Diego Horkis's work, and even Zach Knight, that was the Julius Lab paper. Also, there's a number of people now who've come to define these different cell types, or different enterochromaffin cell types that could interact with neurons below. But definitely Piezo2 is in a certain type of EC cells. And so, yeah, as far as how they interact there, I think that's still wide open. Obviously, the mechanism by which they communicate might be totally different in the gastrointestinal tract. We know like serotonin is really critical as a mediator of communication between cells, and so maybe they function differently there. But yeah, I haven't done any work specifically on the gut cells, but there are a lot of people doing that, and I think it's very exciting, and I think it'll be really interesting to see kind of what those studies reveal.
Yeah, great.
And the gut is really interesting because there's so much mechanical stimulation, right? You have normal digestive reflexes and peristalsis, and there's multiple types of movement in the gut. And so understanding how all of these mechanical forces are activating these cell types, I think will be a big job, but it'll be really interesting.
Great. Yeah. We have a question here from Pascal Roderer, who is kind of following up on one of the follow-up questions that you asked after the first section of the talk. So Pascal asks, do you have a hypothesis for the high-pressure sensor involved in urination? We recently discovered TACAN receptor be involved in this.
Yeah. Well, that's all over. So that that protein is expressed all over the bladder and in neurons, and so yeah, it'll be interesting to see kind of what the role of it is. And there's a lot of other candidates actually. I didn't show this, and now I realize I could have put it in there, but there's Piezo1 in the bladder, and we don't know yet what that's doing. I don't know if that'll be the high sensor, honestly, or the high pressure sensor, because Piezo1, a lot of the times when we see it functioning, it acts, it's in a lot of cells like endothelial cells where it's like more shear stress. And so, I don't know that would be the high pressure sensor. Yeah, but OSCA ion channels were also a recently discovered mechanosensitive group of ion channels. So there's a bunch out there, and we haven't yet done the work to figure out, what that missing piece is. So that, it's a good question. It's actually a huge question, because I'm sure that whatever it is probably the mechanical pain channel, and whether that's DKO or something else, I think time will tell, and there's a lot more work to be done there. So
yeah, it's really interesting to how you're able to parse out the two components of high and low pressure sensing and how they contribute to volitional control.
Yeah, I was pretty surprised, to be honest. I guess there was definitely data that this was the case. People had studied these neuronal responses, so they knew that there are A delta responses and C fiber responses, and so I guess it shouldn't be too surprising, but it was pretty amazing how clear it was to us that only the low pressure ones were missing. And it's hard, when the way I did those experiments, I would manually fill the bladder and we would read out the pressure, and so it's hard to know, physiologically how that differs from natural filling scenarios. And so everything I've shown has been through cystometry, but natural filling might be kind of different. The muscle dynamics are really different in terms of the relaxation of the bladder and stuff. So maybe it's something that we'll understand more about if we can get sort of a natural cycling to work while we're imaging, and that's kind of just a little bit harder because these surgeries take a long time, and
yeah, great. So we have another question here. So great talk. How long does it take mice pups to gain volitional control of urination?
Oh, yeah.
There's plasticity at the urothelium sensory neuron level, or do you think upstream circuits control this?
Yeah, I love the question because mouse pups, I think this person probably knows, and a lot of people know that they can't urinate on their own, so they rely on the mother to lick them. This is true of kittens as well. They'll actually die if they don't get groomed because they can't release urine on their own. And so what's not clear to me is if it's actually the mother's pressure, like the pressure of her tongue that's like forcing the urine out, or if there's some sort of touch-induced reflex. I don't know, maybe someone knows that, but I don't know that that's clear. So yeah, they actually can't urinate by themselves. This is not true of other animals like humans, obviously. Thank goodness we don't have to do that. But yeah, I think mouse pups start urinating on their own around like, I want to say like P10, P15. I don't know the exact day, but sometime in their development, when they're coming up to weaning is when they're able to start urinating on their own. And so, yeah, I don't know where that plasticity is happening. I don't know if it's in the peripheral circuitry, but I kind of, because I'm biased, I sort of think maybe it could be there, that those peripheral circuits aren't really fully developed yet, but yeah, I mean, that's just wide open. I'm just talking, hand waving because I don't really know, but I think it's just a really interesting question, and understanding how these circuits develop will be really cool.
Yeah, it's definitely tractable with the approaches you've developed.
Mm-hmm.
Okay, a couple of questions here from Cesar Mensch, who says, great talk. Are there other piezo-like sensors with different firing thresholds in the same way as you have trip heat sensors that have different temperature thresholds?
Yeah.
And, follow-up question is Piezo2 activation of Piezo2 positive neurons that they innervate the bladder sufficient to induce urination?
Oh, so I haven't done that experiment where we would optogenetically stimulate Piezo2 positive neurons and see if it's sufficient to induce urination. I think that would be really cool, but we have not tried that yet. And urination is complex because ultimately your brain controls it. And so I've talked all about the peripheral nervous system, but in the end, we're actually controlling urination based on context, because you only want to urinate in an appropriate, safe place. And so that's controlled top down through a nucleus called the Barrington's nucleus. And what is unknown as of yet, and is also an interesting question, is how Barringtons, or the cells that mediate those urinary responses, how they receive inputs about mechanical cues, like fullness, for instance. Like, it's not entirely clear how those signals are actually integrated by that system. And it might not be in Barrington's, it might be in different nuclei. But yeah, we don't know anything yet about, so I've just studied sort of locally what's occurring. We don't know how that information gets to the brain or where it's incorporated in the system. And so I think that will be really important to understand. The other question was about different thresholds. And as someone alluded to, Piezo2 has been amazing because it does so many things, but most of those things are innocuous, low threshold things, like touch, for instance. Piezo2, in most cases, does not mediate mechanical pain. So it doesn't mediate, say, like a pinprick response. And so it will mediate mechanical allodynia. That's when touch becomes painful, but that's mediated, again, by touch neurons. Mm-hmm. So yeah, the mechanical pain mechanosensor is still unknown. Takan was published recently. Interestingly, those animals also don't have any pinprick changes in pinprick responses and stuff. So I think there's definitely still an unknown sensor out there, and it'll be really exciting when we figure that out. Piezo1 and Piezo2, if you put them in cells in a dish, they do have slightly different thresholds, I think. But what we've found is that really their threshold is set so much by the cellular environment and the cell type specificity. So actually, Martin Scott from Alex Chesler's lab has a paper on alternative splicing of these ion channels and how that might change their dynamics. For instance, he found like the slow piezo was in the bladder, which kind of makes sense if you think about the stimulus, right? So it's clear that in a given cell type, you'll probably have a lot of other proteins that might change the way that these ion channels respond. They might change the tau of the ion channel or the threshold just based on expression alone. So I think that, in terms of staggering thresholds, it could be due to cellular environment. It's not just the protein alone. cellular environment and other, protein interactions might mediate these changes as well. So I still think there's, a lot more to understand about that.
Yeah, absolutely. So another question here from Du Cheng, who asks, do heart transplant recipients have a loss of baroreception?
Yeah,
if the nerve won't reconnect, is there any work, or would there be any workaround using piezo expression?
Yeah, I'm not an expert in that, but I think that's right. Since they're denervated, they don't have appropriate baroreflex. But so there's also multiple ways to control blood pressure, as I kind of mentioned, where you have acute control and sort of longer term control. So, your body still controls blood pressure by, changing blood volume and changing vasodilation. So, so you still have these like longer term controls, but it's this rapid control that would be missing. And as far as, using piezos to kind of like help, tune different reflexes, I think that would be really cool, but we're still a long way off. It's kind of hard because we don't have any pharmacological tools. So we have one agonist for PIEZO1, not Piezo2. And I mean, if you think about also using a chemical mediator or something to agonize, let's say Piezo2, well, it does so many things in the body that that's going to prove difficult. But yeah, as far as potential therapeutics, I think it's definitely worth exploring if we could localize activity somehow.
Mm-hmm. Yeah. So here, Carlos Quimbedo says, fascinating work and talk. This might be a bit of an odd question, but in engineering, a way of building sensors is to indirectly use mechanical effects to detect other types of inputs, for example, temperature. Are piezo channels used to sense other signals than mechanosensation in the body?
Yeah, so, it's interesting because so far, at least from a chemical perspective, we think that they're really professional mechanosensors and that we haven't found any ligands for them. And in fact, a lot of the things that change or block piezo activity, we think are acting at the protein lipid interface and not necessarily with, the channel itself. So yeah, they're probably affected by voltage, but, to our knowledge, nothing other than mechanical force or tension in the membrane really activates these ion channels. So they're very specific for mechanical force sensing, which is interesting. I mean, I think at some extremes, I'm sure, thermal activity might change their, thresholds or something. But yeah, they really are just mechanosensors, which is also nice because if we see their expression somewhere, we can be fairly certain that that's what they're doing in the cell, that they're not really responding to other stimuli. Yeah,
I mean, it's an interesting idea though that, like, I think there's an idea floating around that Drosophila phototransduction actually is mediated by mechanoreceptors, which sense like stretch. Okay. When light is detected, it stretches the membrane of the photoreceptor cell, and that is detected by mechanoreceptors. So it's involved in the transduction. So it's although it's not piezos. So just the idea of mechanosensation being kind of co-opted to mediate the transduction of others.
Okay. I mean, it's interesting because there's also different, types of mechanotransduction. Like a lot of people who study mechanotransduction study, kind of cellular mechanotransduction, and that, maybe there's a lot of mechanical stimuli in the, surrounding world, right? Or in the tissues around these different cells. And, if you pull anything hard enough, also it'll open. So I think that like, there's a lot of things that are probably affected by mechanical cues in sort of subtle ways. But this is like very specifically gated by tension in the membrane. Yeah. So.
Okay, we'll quickly go through a couple of other questions. Okay, so one here from Julie Agramont. It says, really nice talk. Do you know in which fashion the DRG neurons innervate the bladder? I'm thinking in the way of communication between umbrella cells and DRG. It can be paracrine signaling or a more fast synaptic signaling between these cells. Thanks.
Yeah, the idea right now is that probably it's some sort of paracrine signal. Like I've never seen a synapse-like structure in the bladder, and maybe yeah, I can't think of any examples of that. And, you know, the neuron endings are very different from something like, say, skin endings. In the skin, you have these really specific morphologies where, you know, the neurons wrap around hairs, for instance, or contact directly to Merkel cells. You don't see anything quite so specific in the bladder. So far, I haven't seen anything quite like that. They're more like free nerve endings. And so a lot of people who've studied this, at least studied, you know, what molecules are released by urothelial cells. For instance, again, ATP is sort of the #1 favorite. It's definitely released by these urothelial cells. So it's maybe acting in a paracrine fashion. There are also, you know, P2X receptors on neurons beneath them, and so again, it's sensible that it would work that way. Now, I remain unconvinced that ATP is the mediator downstream of stretch for normal bladder function, because P2X receptors are often nociceptors, and so I wonder if that's actually doing something different than just sort of normal stretch sensing. And maybe in normal stretch sensing, that has a different chemical mediator, a different signal. So yeah, I don't know. I think that remains to be seen.
Yeah. Yeah. Okay. So a question here from George Naratadam, who asks, is there autonomic nervous system dysfunctional in the Piezo mutants? Can cholinergic targeting rescue the phenotype, the Micturition phenotype of the Piezo mutants?
Oh, yeah. I mean, we haven't done that experiment. As far as the autonomic nervous system goes, I mean, basically, so the vagal sensory neurons are mechanically silent. That's all I can say about the double knockouts in the Nodose, for instance. In the animals that we use for the bladder, these are SNS Cree animals or NAV 1.8 Cree. So they lack Piezo2 in DRGs, but only a subset of DRGs, so like 70% of the DRGs, as well as the Nodose. So it actually, that Cree gets both populations. And so, I mean, generally when people think about bladder innervation, they're really only thinking about DRG innervation. There actually might be some Nodose innervation there, or more classic, you know, I think people think of the vagus when they think of like the autonomic nervous system. But yeah, as far as cholinergic output, I mean, I would assume it's normal, but I don't know. We didn't measure that. But yeah, it's interesting because the bladder is one of the few organs that doesn't rely on, say, vagal innervation to function. It really uses DRGs. But the motor portion, like the efferent portion of the nervous system, does seem fine in that, you know, they are ultimately able to urinate. They are able to contract their bladders and, you know, release their urethra. Like all the motor stuff seems okay. So we didn't measure that in a very specific way, but obviously we can see that they're able to contract. So,
yeah. And I'll just sneak in one last question from Yag Kanval, who says, very nice talk and work. Are any piezo sensors present in skin stretch receptor cells?
Yes. So, well, okay. So there's PIEZO1 is in keratinocytes, and there's new data that's out about wound healing and how that might mediate the keratinocyte migration. Really interesting story. And then the neurons in the skin are full of Piezo2. And so we know that Piezo2 mediates most kinds of gentle touch. So not all kinds. It's actually really interesting. And we know this from the human work that Alex's lab has done, where they can't do like 2-point discrimination, but they can sense certain types of gentle stroking. And so if you work from the Ginty lab, for instance, each of these different neuron endings in the skin has a different function. And so, the patients, for instance, were completely insensitive to vibration. So you could put a tuning fork up against their skin, they can't detect that. So it's, probably Meissner's are Piezo2 positive, and Pacinians maybe too. But people actually haven't directly proven that for Pacinians in specific. But yeah, a lot of these ending types are almost certainly Piezo2 positive. And then, yeah, the skin itself, Piezo1. So it's interesting that these ion channels are in different cell types and seem to be doing different things. So,
yeah, great. Well, thanks everyone for submitting such great questions. There were some really interesting ones there. And in the end, it was great that you finished early because we were able to talk for a lot.
Yeah, I actually prefer that.
But having said that, as I mentioned at the beginning, we're going to have a Meet the Speaker session afterwards, and so I hope to see some of you there. All that remains is for me to thank you very much, Cara, for joining us this week. It was a really fantastic talk, and we're very happy to have you.
Thanks.
We're actually going to take a break for a few weeks, so we will be back on March the 22nd with the fantastic John Cryan, who will be talking about his amazing work on the microbiome and its interactions with the gut and the brain. So we hope to see many of you then, and thanks for joining us this week, and thanks again, Cara.
Thanks so much. So I have to, did I say close video? Do you know if that's?
I'll just end the broadcast.
Okay.